Next, we'll hear from Sergio Armando Fuentes Agueda, and we're going to go off to the Gulf of Mexico to learn about the morphological and ecophysiological responses that seagrasses have towards environmental change in the Gulf of Mexico.
Can you hear me?
Yes.
Okay, we're going to start. Just a sec. All right. Very well. You see the presentation?
Looks good. Thank you.
Okay. Now, thank you everyone. Thanks to the society, to the invitation. I will talk about the morphological and ecophysiological responses that seagrasses have towards environmental changes in the Gulf of Mexico. I'm Sergio Armando Fuentes Agueda, a PhD investigator in the Universidad Autónoma Metropolitana-Iztapalapa. So, a little context. Here we have a picture of Murillo of the return of the prodigal son. Why? Because seagrasses are the prodigal sons of the plants. Once they were in the water, they turn in terrestrial plants and they return to the water. That not only gives interesting characteristics of evolutionary, systematic, and physiological to the seagrasses, but also they have an important role in the ecosystems. Seagrasses are, are a key of structure. For example, they are shelter for dolphins and manatee, and also for shrimp and octopus. But also they help to control the dynamic of water. And also they are recycle of nutrients. In fact, they can absorb all the CO2 and control and produce oxygen. That's given. However, I have a bad news. Nowadays, they are threatened. They are reducing the abundance and coverage in all the world. One of the causes is the constructions of anthropogenic activities like constructions and sedimentation of seagrasses. But how seagrasses and climate change work? Well, this is a work of Garcia and Escudero in 2024, and she obtained that, and the collaborators obtained that when peaks of high temperatures rise in specific years, for example, in 2010 and 2021, um, there was a blooming, a lot of flowers in Posidonia oceanica. So she concluded that high temperatures produce high amount of flowers, but reduce the abundance of coverage of seagrasses. Also, if we talk about photosynthetic efficiency, we obtain that seagrasses reduce their photosynthetic efficiency if there is acidification of the oceans and also global warming. As we see in these pictures, if there are high temperatures in Sostera noti, the leaves are dying off, and also acidification, that is an acid pH. Here we have the distribution of Thalassia testudinium. Thalassia testudinium is distributed in the Caribbean Sea and in the Gulf of Mexico. The objective of the talk is to share some of our findings in seagrasses meadows in the Gulf of Mexico during 4 years. We will cover 4 years, seasons dry, rainy, and nortes. We obtain biomass, environmental parameters, epiphytic, phytoplanktonic, and photosynthetic efficiency. This is the study area. We have 6 areas. The first one is Laguna de Términos. The second one is Champoton. The third one is Petenes. The fourth one is Silam, San Felipe, Yalajao, and Cabo Catoche. There were monitoring for years in the seasons dry, rainy, and nortes. And this is the coverage. We obtained a total of 3,375 kilometers square of seagrasses in all our area. The highest area is the complex Campeche Petene Solestun, and the lower area is Cabo Catoche of seagrasses. This is thanks to a eco-sounder that it was monitoring in these 4 years all the area. The environmental parameters we obtain salinity, temperature, pH, dissolved oxygen, and depth, and column waters and pore water nutrients. Also, we obtain the biomass of Thalassia testudinum and flowering, and we number of flowers and how is the biomass of Thalassia, and then the biomass of epiphytes and phytoplankton. We obtain the chlorophyll analysis using spectrophotometer with Strickland Parsons and score UNESCO. And the photosynthetic efficiency we obtain with the fluorometer in plants. And what this means? Well, point 7 means that the 70% of the light will be available in the photosystem 2 in all the plants. This is the value of photosynthetic efficiency, or FVFM. We obtain Wallis analysis for all the parameters and a principal comment analysis for environmental parameters. So, what we obtain? We obtain that there are peaks of high temperatures in the rainy season. However, since 2018, there is a high amount of high temperatures, not only in the rainy season, also in the norte season and in the dry season. And in the rainy season, it will be high temperature. The acidification of the ocean, we obtain that in the rainy season is acid. However, at the end of the monitoring, it will be stable. Also, we obtain that the salinity, there are cycles. In dry season will be high salinities. So we can conclude, in this part, we can say that in the 4 years, there is a climate change. However, also we find differences in the areas. Area 1 and 2, that is Laguna Terminosa Chapoton, and Laguna Yalajao and Cabo Catoche, are high in temperature. And also we obtain that pH is high in the area 5, that is San Felipe. Also, we obtain that Champoton has the highest depth, and the lowest depth is in Silam. The principal content analysis, we obtain that the factor 1 is composed by pH, salinity, and temperature. The second factor is obtained with nutrients, that is total phosphorus, nitrogen, and silicates, and also depth. The biological differences, we obtain that the highest amount is the area 4, that is Silam, and Cabo Catoche are the highest of biomass of Thalassia testudinum. However, the flowers were higher in the complex Petenes that has 44.63% of flowers. In the phytoplankton, we obtained that the area 5, that is San Felipe, has high amount of phytoplanktonic biomass. The epiphytes were higher in the lagoons, that is Terminos and the Laguna Yalajao. And the photosynthetic efficiency we obtained, that is higher, 0.70% of the light were higher in all the sites, except San Felipe, that is lower than 0.60. So we can conclude that in San Felipe, all the plants of Thalassia testudinum will be in stress. We do a PCA, and we obtain that the area that is separate with all the areas is the area of Laguna de Yalajao. We can speculate that this area has different conditions than all the areas. So, what mean our results? First of all, there is a climate change, and how seagrasses are affected by climate change. First of all, if there are high temperatures, there will be losses of leaves in Thalassia testudinum. Also, high salinities, higher depths, and limited in nutrients. In our case, we are not limited in nutrients. In fact, in the rainy season will be higher nutrients because in all the peninsula of Yucatan, we have 3 seasons. Dry season will have high amounts of salinity, and however, the rainy season will be high amounts of temperature and lower pH and high amount of nutrients, because all the rain and all the fresh water will apport in these environment parameters, and the north season will be high dynamics of water. How seagrasses are resilient to climate change? First of all, if we have high amounts of seagrasses, it will control the pH. It will reverse the acidification of oceans, in where the literature said. Also, the optimum temperature is 25 centimeters to 35, that propose to have a healthy environment. So in our case, we can say that there is a climate change, but depending of the areas will be the impact of the, for example, if we have high amount of nutrients and we will have high amount of phytoplankton biomass and high temperatures. Also, will we increase the macroalgae in the area and reduce the seagrasses? This is an event that is happening in the Mediterranean. Our data suggests that there are areas that are vulnerable. Not only there are amount of high anthropogenic activities, but also they are reducing of the mangroves and there is high amount of macroalgae. For example, in Laguna de Terminos, in Silam and San Felipe, and part of the Laguna de Yalajao. There are anthropogenic activities that reduce seagrass coverage and abundance. For example, it has been reported that they are destroying part of the San Felipe, Yucatan for constructing houses or complex for living. This is coherent because our observations have seen that there are a lot of macroalgae and the photosynthetic efficiency is lower than all the areas. So we can say that San Felipe is a high vulnerability area for seagrasses. However, we have natural areas that resist the climate change. For example, in Petenes, not only has high amount of nutrients, also high amount of nutrients, but also it will increase the biomass of Thalassia and increase the number of flowers. Although Cabo Catoche has a lot of biomass, it can produce a lot of flowers than in Petenes. So natural areas will resist more the climate change. To conclude, we said there is evidence that the Peninsula Yucatan has climate change. Seagrasses are threatened by climate change. However, conserved areas will resist the climate change. Laguna Terminal, San Felipe, and Yalajao are threatened by anthropogenic activities like construction and sedimentation. I want to thank the one Seagrass laboratory staff. We have the head researcher, that is Margarita Gallegos. Also, we have some members of the staff that help not only for sampling, but also for analyzing and studying this information. And we have our students that are working with their data and their papers. Thank you very much.
Thank you, Sergio. Please put your questions in the chat for him, and now we'll get to addressing those questions from all of our speakers. I'll start off with Nick. Both Nick and Caleb have started responding to questions in the chat, so I'll aim at those that haven't been responded yet. Nick, can you speculate on the role of the seed bank on any lags?
Yeah, absolutely. So prior to about 3 or 4 years ago, when we saw the extirpated population actually come back after a couple of years, we didn't know that a seed bank actually existed. It was highly debatable in Guttatus. I think there are two things that a seed bank can do, right? They can help prevent your population from going extirpated permanently, and it can also create legs, right, that allow evolutionary rescue to slow. In our case, we had one introduction event, right? And then we keep on seeing seeds come up from that single introduction event, we think too. So we think that this actually helps our assisted gene flow because you're initially introducing those alleles at such low frequencies that genetic drift will make them leave the population fairly frequently. So you're getting multiple chances from a single introduction event with a seed bank. Those are kind of my broad thoughts on seed banks. And I don't think we know nearly enough about seed banks for any of our species.
Thank you. Next question for Nick. Based on your results, it seems that AGF techniques can sometimes feel like we're playing with dice, especially when given the many interactions that need to be considered. How would you recommend approaching this type of research for a species with a very limited number of individuals and those on the brink of extinction?
Yeah, I left that one on purpose because I would think that's a great discussion topic to have actually up there. So for the thing is, we need model species and we need research for the some model species to kind of think about how we may want to do this in limited or threatened populations. I don't think we have nearly enough of that yet. But I think in these really limited populations, we might want to take the approach of some of the animal researchers out there. So for instance, Hawaiian tree snails, right? They have gone extirpated in huge numbers due to the introduction of, rosy wolf snails out there. But they've taken them into breeding facilities figured out what was going on in increasing genetic diversity in the lab, and then brought them back out to natural populations. So I think by using some of these kind of more targeted approaches, we're actually bringing things in, that might be a really helpful way to go about it.
Thanks. And last question that I have, excuse me, for you, Nick, is for the species with mixed mating, like Mimulus guttatus, I wonder to what extent introduced populations rely more on selfing for reproduction.
Oh yeah, that's good too. So I think that, Mimulus historically has been an outcrossing species, even though it is capable of being self-compatible. In the field, we mostly see outcrossing occurring. But in those high elevation populations, we think there's becoming mismatches with pollinators up there, and reproductive assurance is becoming more and more important in these populations. So if those California populations tend to be even more outcrossing than the Oregon populations, well, one, that might bode well for getting alleles into those populations. But on the other hand, it might not bode well for evolutionary rescue because you have a different mating system than what's actually selected there. So that's two things that could be button heads and preventing assisted gene flow from being effective.
Thank you. I'll switch to some of the questions, Caleb, that I might have missed if you got to. And for all of our speakers, kudos for your talks. Any thoughts on biotic homogenization between the mountaintops due to climate change?
Yeah, no, that's a great question, and it's an awesome topic. I think that yeah, we'd love to like specifically look at, like, for example, changes in beta diversity over time, and I think changes in turnover over time, which we sort of like briefly explored for this manuscript, kind of left, partly just to, I really wanted to make this manuscript as like intuitive as possible, right? So like, what's the change in species richness? What can we say about lags by combining that with turnover? Like, how can this sort of be the biggest possible scale, and also like how can we like quantify, like, what's the baseline amount of turnover that we would expect in these communities? That was like a question that I really grappled with it in this, was like, is 21% like an insane amount of turnover, right? That means like a fifth of on average across all this pairs of sequential surveys, like a fifth of the species between any two surveys are going to be different. So yeah, we did observe a lot of turnover, but whether or not that turnover, I think we use the null model to try to say that turnover isn't like directed towards like more warm species or more of a certain species and less of a different and less of something else. So yeah, we haven't looked at biotic homogenization exactly, but yeah, my sense is that things have changed not a lot. And so maybe we're not seeing biotic homogenization now, but we could.
That was actually my question. My thought was generally towards losing rare species at those high elevation sites and favoring generalist species up there.
Yeah.
So you see with invasive species as well, fairly frequently.
Right. Yeah, and I think actually we found that like partly what's driving that sort of what seems like a lot of turnover is we try to time our surveys to go to these peaks at the height of the growing season, but then the height of the growing season looks different in different years. For example, like on those desert peaks, like we're in Death Valley, and there are years where there's good years where you see like all of the geophytes and ephemeral species and those kind of rare from that perspective. And then years where you don't. And so I think actually some of those like rarer, more ephemeral, like difficult to detect species are driving increases in richness and increases in turnover. Yeah, at least over 4 surveys, which in our case is 2 decades.
Okay. And one more question for Caleb here. You capped your talk by saying generally there were limited plant community changes across all mountain ranges in the study over 20 years. What does it look like when you just zoom in on the mountaintop that fall at or near the subalpine or tree line ecotone?
Yeah, that's really cool. I haven't like just pulled out like, okay, if you just look across all the lowest elevation peaks, and yeah, there's sort of this like inherent elevation gradient, at least towards summits of different elevations inside of this. So I haven't like just pull. I think that's a great idea, and that's a really fun. Maybe that's what I do during my lunch break today. Is just like pull out all the lowest elevate or all the peaks that are at the lowest in every single target region and think about those changes. I think that they haven't changed a lot, or that I don't think you would see a completely different pattern by doing that. But I like that idea, and I think that's the idea that we're moving towards is sort of where in the Alpine, at what elevations would you see the fastest rates of change or the slowest rates of change?
Okay, you can get back to us at the end of the session. Just kidding. Um, so a few questions for Sergio. How many of your studies had overlapped with mangrove habitat? Are there any general trends or observations that could be made with mangrove seagrass interactions in that area?
Well, there are no many works for interaction of seagrass and mangrove. What we obtain is the vulnerability map was obtained by the mapping, the areas and the loss of mangroves. But the hypothesis is that first the seagrasses recycle nutrients that help the mangroves to grow, and also they help to control the dynamic of water. For example, if there is a hurricane, they can control the impact of the hurricanes and reduce the losses of mangroves. But it's interesting to study the interaction of mangroves and seagrasses. That could be interesting.
Okay, I'll ask one more question so we can stay on time. Sergio, many of your sites had overlap. Wait, sorry, read that one. Moving up. Encouraging to see that reducing human disturbance could lead to the survivability of seagrass. Could calcareous macroalgae also help reduce acidification in disturbed areas?
Yes, the macroalgae. However, the problem for macroalgae is that they are like they are what can I say? They are not like species that are long term. They're short, maximum like 1 year of life. So the impact is lower than the seagrasses like Thalasseas tetudidium. But the diversity in Petenesi have a high diversity of macroalgae, so it could help also the acidification.
Okay. And I will squeeze in one more. What were the parameters that you took to say this is due to climate change?
Well, first of all is the the epiphytes. First of all is the phytoplanktonic parameters that increase in some areas. For example, in San Felipe that is higher. The community of of these microorganisms increase with high temperatures and high amount of nutrients. So climate change, if temperatures are higher, these microorganisms will increase, the phytoplanktonics will increase and reduce the coverage of seagrasses that we see in San Felipe. Maybe it's not all the area, but specific areas that are not conserved will be affected with climate change.
Thank you. And there are questions, I think, for each of you still in the chat that we didn't get to, but it's time to move on to our next
Okay.
session. Thank you all.